Neutrophils are the most numerous immune cells in the human circulation and are quickly recruited to sites of infection or inflammation, forming the first line of immune defense. When the intestinal barrier is damaged, neutrophils are recruited from the circulation to the inflamed tissue through a plethora chemotactic gradients formed by cytokines such as IL-1 beta, IL-6, and tumor necrosis factor, or TNF, alpha. Chemokines such as CXCL8, chemokine CXC motif ligand CXCL10, and macrophage inflammatory protein 2, MIP2, also known as CXCL2, and growth factors such as granulocyte-macrophage colony-stimulating factor, GM-CSF, and granulocyte colony-stimulating factor, G-CSF. Neutrophil recruitment is also mediated by bacteria-derived molecules such as formyl-methionyl-leucyl-phenylalanine, FMLP, and short-chain fatty acids, SCFAs.
In healthy intestine, left, damage to the intestinal barrier triggers the recruitment of neutrophils from the circulation to the inflamed tissue along a chemotactic gradient formed by cytokines, IL-1 beta, IL-6, TNF-alpha, chemokines, CCL8, CXCL10, MIP2, and growth factors, GM-CSF, and G-CSF. Neutrophil recruitment is also mediated by bacteria-derived molecules such as formyl-methionyl-leucyl-phenylalanine, FMLP, and short-chain fatty acids, SCFAs. The participation of neutrophils and nets in IBD is a double-edged sword. Right. Neutrophils cooperate in wound healing and the resolution of inflammation by releasing vascular endothelial growth factors, VEGFs, and lipid mediators, protectin D1 and resolvin E1. These factors impede neutrophil recruitment and promote phagocytosis. Nets impede the spread of microorganisms by trapping them in an environment of microbicidal components and stimulate the healing of the intestinal mucosa. Neutrophils directly cause tissue damage by releasing neutrophil elastase, proteases, MMPs, pro-inflammatory cytokines, IL-8, TNF-alpha, IL-1 beta, leukotriene B4, and ROS. These factors provoke not only injury to the epithelial barrier, but also the recruitment of neutrophils and other immune cells to the inflamed tissue. Neutrophil recruitment is also promoted by cytokines IL-1 alpha, IL-17, IL-22, G-CSF, and GM-CSF. Lack of the IBD protective gene CARD9 in neutrophils enhances ROS generation. IL-8, TNF-alpha, and PAD4, increased in UC patients, contribute to net production. Accumulation of nets in the colon is accompanied by the induction of tissue damage and inflammation, as nets also boost TNF-alpha and IL-1 beta production,